Okay, so today's topic is beta oxidation pathway. Okay, now first of all you should understand that beta oxidation is actually destruction of fatty acids for gaining ATP. Okay, that is the main aim is to oxidize fats and to get energy out of that. That's our first aim. Okay, now we will discuss beta oxidation pathway step by step. Okay, starting from our diet, how we get fat, and then how we can transform this fat into monoacyl glycerol. Then from monoacyl glycerol, how we convert that monoacyl glycerol again into tag, that is triacyl glycerol. Then after that we will talk about the oxidation of that fat, tag, and after that then we will talk about beta oxidation pathway. Now listen, when we are talking about beta oxidation pathway, you should always understand that it all starts from gastrointestinal system. Okay, it all starts from gastrointestinal system. Now listen, for beta oxidation pathway there are many things which are involved right now. We'll discuss it step by step. Listen, the first thing we need is triacyl glycerol or tag. Okay, now what is tag? I have already discussed this in your lipids video, introduction to lipids. That is, tag is actually made up of two things. Okay, right? That is, number one is glycerol. Number two is fatty acids. Now, what do you mean by the term fatty acids? Listen, fatty acids are carboxylic acids. Okay, fatty acids are carboxylic acids which have carbon number 12 or more than 12. Okay, or more than 12. That means those fatty acids which have carbon number more than 12, they will be considered as the real players in beta oxidation. Now listen, first of all you should understand that this is the structure of glycerol, right? I have already discussed it in great details when we were studying introduction to carbohydrate. Then we have moved to metabolism, right? Now listen, now this is the structure of glycerol, and suppose, what do you mean by fatty acids? Now listen, how to draw a fatty acid I have already discussed, but still we need some recap. Now this is the R group, this is the CH2. Okay, now NCOOH, this is the safest way to draw any fatty acid, right? Now listen, this COOH will now react with this OH, that means okay. Okay, now listen, this is the overall structure of a triacyl glycerol, or which is also known by the name of tag. Okay, this is overall the structure of triacyl glycerol or tag. Now listen, you should always understand that these notations, that is, this is how we draw phospholipid or something like that. That is this we draw one circle and we draw these two lines, these two things. Now you should always understand what do you mean by these things. Okay, these are the fatty acid chains. These are the fatty acid chains. Okay, these are the fatty acid chains, and this is the glycerol head. Okay, this is the glycerol head. Now listen with all your attention. You should always understand this is the general structure of a phospholipid, right? Because here one phosphate containing group is present. But in tag, how we can draw a tag, that is triacyl glycerol? We will have to draw a tag like this. That means there are 3 legs in tag. There are 3 legs. Each leg corresponds to one fatty acid side chain, and this head actually, this head is nothing but this glycerol molecule. Okay, this head is nothing but this glycerol molecule. Okay, now we should now understand that we should always understand that we are talking about beta oxidation of fatty acid, right? Now listen, when we are talking about beta oxidation of fatty acids, that means we are talking about the beta oxidation of these chains, not this. That means we will have to exclude this part and we will have to use these parts to extract ATP. Okay, we will have to use this part to give rise to ATP. Now, you should always understand one thing, that what happens to this glycerol part, right? Now, this glycerol is converted actually to pyruvate. Okay, glycerol is converted actually to pyruvate, and from pyruvate we know we can always convert this pyruvate to acetyl CoA, right? And this acetyl CoA will enter Krebs cycle. Understand this very basic thing. Okay, that is what happens to these two portions of fatty acids, of triacyl glycerol or tags, or which is also known by the name of triglycerides. Okay, the fatty acid chains are converted to ATP via beta oxidation. Okay, they can also be used in other ways. Now, the most important thing you need to understand here is what happens to this glycerol part. This glycerol part is first converted to glycerol 3 phosphate, then it is converted to pyruvate, and from pyruvate we can convert it to acetyl CoA, and this acetyl CoA enters Krebs cycle to give us again ATP via electron transport system. Clear? Now, we should again understand these basic things, that is, suppose we are in diet. Okay, suppose we have taken a heavy diet, and in the diet triacyl glycerol is present. Now, in the triacyl glycerol, you should always understand that there are 3 acyl part. These are the acyl parts. Okay, these are the acyl parts. Now, there are 3 acyl parts, therefore they are known by the name of triacyl, and there is one glycerol, that's why glycerol. Now, important part is this, you need to understand that there are 3 acyl groups and there is one glycerol molecule, right? Now, you should always understand that there is no mechanism in our body by which a whole tag molecule can be absorbed. Okay, tag is generally absorbed in the small intestine, but there is no direct mechanism by which tag is absorbed into our body. There is no direct mechanism. So what is the basic mechanism by which tag is absorbed in our body? Now listen. Now you should always understand. Now listen. Suppose this is your GI tract and this is your small intestine, suppose, and suppose this is your small intestinal cell. Now listen, in the small intestine tag will be absorbed. Okay, now go to my lipid absorption video, you will understand how lipids are absorbed. Okay, now we will only talking with respect to tag only. Now why I am talking about all the lipids, because in our body not only tag is present, cholesterol, mag, DAG, diacylglycerol, monoacylglycerol, all are present. Now listen, whenever we are talking about tag, you should always understand, that tag is having 3 acyl groups and 1 glycerol molecule. That's why it's a big molecule. Now it cannot be transported directly into the cell. So how does this occur? Listen, tag is at first converted into monoacylglycerol and 2 acyl groups. That means lipase, pancreatic lipase, there are pancreatic lipase. Now listen, pancreatic lipase converts tag into, actually breaks down tag into 2 acyl groups and 1 monoacylglycerol. What is the structure of monoacylglycerol? Now listen, suppose this is your tag. Okay, there are 3 legs. Now listen with all your attention, there are 3 legs, right? Now my question is, suppose you have put some lipase here. Okay, we are not talking in details about the absorption of lipid, okay, because there are many factors called colipases. Okay, now listen, now suppose you have attacked this particular tag with lipase. Now first things first, suppose there are 3 acyl groups. This is number 1, this is number 2, this is number 3. Which acyl groups will be removed? Which acyl group will be removed? You are telling first, 4th, 1, 2, 3, all the 2 and 3. Okay, he is telling, oh, SU and you are SA. Now listen, all of you are wrong. Thank you. Okay, now listen, only 1 and 3 will be removed. Okay, only 1 and 3 will be removed, and this 2, oh my god, it is a strange thing here, but we will not discuss about these things. Okay, listen, so this is known by the name of 2-monoacylglycerol. This is the correct name of this. Okay, 2-monoacylglycerol. Okay, somebody is showing something bad, so let me not disturb your senses, which are very strong towards these things. Okay, now listen, that means you need to understand that this is actually one molecule of 2-monoglycerol. This is 1 into 2-monoglycerol and 2 into acyl groups. Okay, now what will happen is this monoacylglycerol will find its way into the cell. This 2-monoacylglycerol which will enter the cell. Okay, now a very interesting thing happens, that is, within the cell this monoacylglycerol gets two fatty acids and it is again converted into tag. Okay, it is again converted into tag. Now these tags, many tags, there are many tags outside and therefore there will be many tags inside, and these tags, triacylglycerol, will form a structure which can easily diffuse through the cell membrane, known by the name of chylomicrons. I have already discussed this in great details when I was discussing lipid absorption, chylomicrons. Okay. Now this chylomicrons is actually, what is the use of chylomicrons? Chylomicron is like a boat, you know. In blood the main constituent, the main matrix is plasma, right? We have 55% plasma in our blood and 45% blood cells, right? Now that is the main constituent of our blood is plasma. Now the point you need to understand is this: plasma is mainly, though plasma is a colloid, but it is mainly made up of water, right, and proteins. Now listen, what is the similarity between water and proteins? Both are polar. Tag on the other hand is non-polar, right? So what is a chylomicron? Chylomicron is like a boat, okay? It's like a boat. Now listen, chylomicron is actually like this, that is, it's a lipid bilayer. It's a lipid bilayer. Now what is a chylomicron? Chylomicron is a big lipid bilayer. Okay, it's a big lipid bilayer. Okay, so chylomicron is like a lipid bilayer. Okay, now the first question is this: suppose the chylomicron is moving through a blood vessel. Suppose this is a blood vessel. Okay, now when I said that tags cannot travel through plasma, therefore it is taking the help of chylomicrons, and now I have said that chylomicron is a lipid bilayer. First point that should come into your mind is how a lipid bilayer will help in the transport of tag, because tag is also what? Non-polar. Lipid bilayer is also non-polar, but you don't know one thing, that is this lipid bilayer is loaded with some proteins known by the name of apolipoproteins. This lipid bilayer is having some proteins known by the name of apolipoproteins. And chylomicron have one type of apolipoprotein known by the name of ApoB48. Chylomicron have one type of apolipoprotein, is one of the type of, there are many type of apolipoproteins. Okay, now one of the type of apolipoprotein that chylomicron have is ApoB48. Okay, now listen, now this is the actual thing. How will you distinguish chylomicron from other lipoproteins? Chylomicron is one of the lipoproteins, right? Why it is called lipoproteins? Because it has got proteins in the form of apolipoprotein and it is loaded with lipids. That's why apolipoproteins. Now listen, sorry, that's why it is known as lipoproteins. Now listen, first things first. Now inside it there are tags, there are cholesterol. Both are non-polar. For their transport across this polar medium, they need a polar environment around them, and that's why apolipoprotein is helping in that. Okay. So what we have actually achieved till now is we have taken our tag through diet. We have taken our tags through diet, and we have successfully converted or digested tag into monoacylglycerol and acyl groups, and this monoacylglycerol now enters this cell which is known by the name of intestinal cell. Okay, it is intestinal cell. Now listen, this MAG now enters intestinal cell and it is again converted inside the intestinal cell into tag, and these tags are loaded onto chylomicrons, which is a phospholipid bilayer having apolipoproteins over it, on the surface of it. Why? So that it can easily swim through plasma, whose main constituent is water, and water is a polar medium. And as apolipoprotein is a protein, therefore it is also polar, and polar likes to dissolve in polar medium, like dissolves like. Now that means up to this stage what we have done is we have not even started deoxidation. Now what we are actually trying to do is, before the start of beta oxidation, it will take maximum of 15 minutes. But you should understand that how these things are going on, because if you don't have tag, if you don't have these receptors through which MAGs enter, then you will have some very big problems in your body. Okay. Now listen. Now this tag loaded chylomicrons will enter circulation. These tag loaded chylomicrons will enter circulation. Okay. And what will happen? Now listen, there are few cells which love to take triacyl glycerol. There are few cells which love to take this triacyl glycerol. What are those? Adipocytes. Very good. And liver. Okay. Mammary glands. Okay. And brain. Brain don't use much fat. One of the best source of lipids are your muscles. Muscles take up loads and loads of tags. Muscles takes loads and loads of fat. Why? There is an interesting point right now. Listen. So these tag loaded chylomicrons have now entered the circulation. Okay. Now what will happen is, now suppose this is whenever I draw a cell with a marginalized nucleus and a large cytoplasm, it is known by the name of adipocyte. Now first I will talk about how these tags which are loaded inside the chylomicrons are taken into the cytoplasm of adipocyte. Why I am talking about adipocyte? Because they are the masters of taking tags in our body. And believe me or not, if you understand this mechanism, then you will probably understand the mechanism of all cells which take up tags from these chylomicrons. They are similar. Listen. There is an enzyme. There is an enzyme waiting for these chylomicrons to come. You know what is the name of this enzyme? What is the name of this enzyme? What is the name of this enzyme? If I tell you, suppose if I tell you that these enzymes, these are surface enzymes, okay, which are attached with the cell outer portion of the cell membrane of adipocytes. Now listen, if I tell you that these cell membrane attached enzymes react with chylomicrons and they take up what? Suppose this is your chylomicron and inside it is tag. Now if I tell you one thing, that whenever chylomicrons interacts with this enzyme, tags will be broken down. Okay, tags will be broken down and will enter as MAG and acyl groups. Can you name this part? Can you name this enzyme? Whenever chylomicrons interacts with this particular enzyme or protein, this protein or enzyme converts or breaks down tag into MAGs and acyl groups. No, this is not hydrolase. This is known by the name of lipoprotein lipase. Lipoprotein lipase. Okay, this is the name of LPL. That means lipoprotein lipases are enzymes. Lipoprotein lipases are enzymes which are present on the surface of adipocytes or any cells which are master in taking tags from these chylomicrons. That means lipoprotein lipase are enzymes Listen with all your attention. Lipoprotein lipases are enzymes which are present on the surface of adipocytes or any cells, which are masters in taking tags from cholesterol to those cells. Okay, those cells means those which actively require tags. Okay, now listen with all your attention possible. We have got MAG, right? We have got MAG here, that is monoacyl glycerol, you know, 2 monoacyl glycerol. Now we have also got acyl groups here.
Well, in that monoacyl glycerol, it's directly transported as such through chylomicrons.
What?
We have MSG at intestinal cells, and they can enter the bloodstream as through chylomicrons and get transported here.
Okay. She has raised a beautiful question, that is why we need to convert this 2 MAG into tag. Then you know we will have to transfer this TAG via chylomicron into these cells, where we ultimately need MAG, right? Now listen with all your attention. This question has to be answered. Listen. Okay. You're raising this question because you probably don't have much base in biochemistry. That's the true fact. Listen. Whenever, suppose this is your glycerol molecule, this is your glycerol molecule, okay, and these are your fatty acid chains. These are your fatty acid chains. Now you need to understand one basic thing, that when you remove these groups, okay, when you remove these groups, this becomes polar. That means MAG is very good for cytoplasm, because cytoplasm is also made up of water. But the point you need to understand, why we need to convert MAG into TAG or tag is this, that chylomicrons not only transfer tags, they transfer tags together with cholesterol, and cholesterol is a non-polar thing. That's why we cannot combine MAG with cholesterol, because they will react, because they are highly polar. That's why we will have to convert this into tag, so that we can pack tag and cholesterol together, so that they won't interact with each other. Okay, now that's why our body has made a system, a beautiful system, using which we can convert that tag into MAG again. We need this MAG in these cells. Now listen another thing. If we just only transfer this MAG here, what will happen? Listen. From this MAG, further 2 acyl groups which are already produced by this cell will attach, and they will form tag. But another thing won't happen here. Why? What do you know? That the 2 acyl groups which were released by this tag, these 2 acyl groups will actually undergo beta oxidation. These 2 acyl groups are actually needed for beta oxidation. Okay. Now that means you need to understand that where beta oxidation is taking place. The first thing you need to understand about beta oxidation is this: these acyl groups are actually oxidized to produce ATP. These acyl groups are actually used for the production of ATP. Now listen. Okay, so now we have already finished how these acyl groups reaches the cell. Now we will talk about the beta oxidation proper. Okay. Listen. suppose this is an adipocyte and we have a marginalized nucleus. Now, again you should always understand that here is lipoprotein lipase LPL, right? Now, we have already got monoacyl glycerol or 2 monoacyl glycerol and 2 acyl groups. Okay. Now, you know where beta oxidation takes place? No, that is a cellular. No, listen. Okay, yes, listen. If I tell you where beta oxidation takes place, there are 2 places: extracellular and intracellular. If I tell you that in which cell it takes place, that is mitochondria, muscle, first thing that should come into your mind when I am talking about the place or the cell in which beta oxidation takes place, muscle, adipocyte, these 2 cells are enough. But whenever talking about the intracellular compartment of beta oxidation, you should always understand the proper answer is mitochondria. Now, why mitochondria? Why mitochondria?
It has all the enzyme required for that.
You know, there is one theory known by the name of endosymbiotic theory. In endosymbiotic theory, what we have learnt is that mitochondria was actually a bacteria which was living outside the cell, but as the atmosphere of Earth changed, it actually entered the cell and it actually had a symbiotic relationship with the cell. How can a bacteria have much more sophisticated enzyme than us? That's my question. Listen, they don't have the enzymes. In most of the cases, they don't have the enzymes for beta oxidation. They took help from our own DNA and they produced those enzymes. But why our cell keeps this enzyme in the mitochondria? Because mitochondria have proper pH so that these proteins or enzymes won't degrade. That's the only reason. Okay.